Hi, good day, everyone. In this short video, I'll be talking about isolation of protein. How do we isolate a protein from a cell and purify it? Now, it depends on the cell we want to isolate a protein. Which cell? It's a cell from a prokaryotic cell organism or a cell from a eukaryotic organism. If it is from the eukaryotic organism, is it a cell from an animal cell? Sorry, a protein from an animal cell or a protein from a plant cell? So in either cases, what is most important is that where is this protein located? So protein can be located inside the cytosol. It can also be found inside the organelles. So for you to be able to isolate these proteins, you need to break open the cells. If the proteins are found within the cytosol, then you extract the cytosol content. You collect the cytosol content. And then from there, you know that your proteins is there. And discuss the remnants, the organelles, the other organelles, including the remnant of the something. But if your protein is found within the any of the organelles, then you have to isolate these organelles. So first of all, now starting with the breaking open, to break open the cells. How do we break open the cells? We have basically two ways, either using a chemical method, where we use saponin, or SDS, sodium dodecyl sulfate. And these chemicals, what they basically do is when you added these chemicals to a test containing cells, what basically they do is they interact with the membrane lipids. And once they interact with the membrane lipids, they cause a distortion in the membrane lipids, causing the breakage or opening of these membrane lipids. Therefore, the content will be released out. So when this happens, you have the cells, also the organelles being suspended out. You have the membrane or the plasma membrane being broken down using a chemical. But there is another way also mechanically. You can freeze and thaw. Freezing and thawing involve freezing at high temperature until the cells got frozen, and then suddenly raise the temperature up so that they get cooled up faster and freeze again. Got cooler faster spontaneously. This can cause the breakage of the cells. The cells will get broken open, and then will break open, and then you have the content to get released. Or you can use a homogenizer. You can use mortar and pestle to homogenize, to break open the cells. So when you have this happening, what is the most important thing now is to determine where is the protein located. So if your protein is located in the cytosol, then you can centrifuge your lysate, the crude extract. When you centrifuge your crude extract or the cell lysate at 1000 G, you will have your sulfonatant, and you will have some component found as a pellet. But you can actually spin more than at least more than like around the 80,000. The 80,000, you know that all the component, all the organelles must have been pelated or must have moved down in form of pellets or settled down as a pellet. Then what you will have is the component of your cytoplasm. And in cytoplasm, you have your protein suspended there. So you can collect it and then describe the pellet. But if you're interested in a protein located in a particular organelle, then you can carry out either differential centrifugation, where you centrifuge. Post centrifugation, remove the larger organelles like the nucleus, like the mitochondria, go to the second one at around the 20,000, get the next one, like this, until when you got the organelle of interest at the pellet, then you collect it, break it open also, So that what you have, are the components that are found that organ specifically. So all the proteins we are there. If you're interested in a particular protein, the next thing you will do is okay, so this is the differential centrifugation where you centrifuge at 1000 G to get your nucleus, to get cytoskeletons, to get plasma membrane, the broken plasma membrane, uh, nucleus. And when you centrifuge at 20,000 G for 20 minutes, you will get mitochondria, lysosomes, peroxisomes. When you centrifuge at 80,000, you get all those macro somes like endoplasmic reticulums and other small vesicles, because etc. And when you centrifuge that at about 150,000 G, you will have the content in the smaller, like the ribosomes and other large macromolecules that are suspended inside. So you can have this as your proteins if you're actually interested in this component. And then from there, you proceed. Or you do what we call density gradient separation, where you prepare a sucrose of a different concentration, and then you layer them inside the test tube, and then you put your samples. So those samples will be separated based on the size, so with the smaller ones being at the bottom, at the top, and then the bigger ones at the bottom, because they are heavy enough to be able to pass through the much denser solution of sucrose. So you have quite a number of reactions containing a similar set of molecules, or molecules of the same molecular size. So the next thing is what we call salting out, or ammonium sulfate precipitation. Remember, I told you how a protein become a soluble in a solution. What made a protein to be soluble in a solution is through the interaction of the ions inside the water. So the ions, water forms these ions, which are hydroxyl ion and hydroxonium ion. So if you have a molecule that can actually form a partial interaction with this hydroxyl and hydroxonium ion in a water, that molecule can potentially become soluble, or can potentially dissolve in water. But if a molecule could not be able to form this potential interaction with the hydroxyl ion and the hydroxonium ion inside water, or formed by water, that molecule can not dissolve in a water. So in the case of the proteins, we talked about that proteins are made of amino acids. And these amino acids, there are hydrophobic amino acids, there are hydrophilic amino acids. The hydrophilic or polar amino acids comprise of all these amino acids that contain a polar groups, and including those amino acids that are charged. We have positively charged amino acids, we have negatively charged amino acids. So these amino acids are the amino acids that contribute to polarity, that contribute to solubility of the protein in a solution. So because of this, these proteins were able to get to be in a, or to interact with the surrounding polar environment inside the water, or to be found inside the water, being soluble inside the water. That is what is preventing them from precipitating out. But remember, the hydrophobic amino acid here, you have a protein in the folded form. So the hydrophobic amino acid in most cases are not positioned on the surface. They are somewhere embedded inside the protein, so they don't interact with the surrounding aqueous environment. So now, how do you precipitate the protein out? Remember, this potential interaction between the water molecule and this polar group of the amino acids, you kind of interfere with this interaction. How do you interfere with this interaction? One of the methods is to add ammonium sulfate. An ammonium sulfate in the solution, what happened to it? It dissociates to form an ammonium ion. The ammonium ion is a positively charged, and the sulfate ion, which is a negatively charged. Now, the ionic strength of these ions compared to the ions produced inside the water, ammonium ion and the sulfate ion, are more stronger. They have more strength, and as such, they can easily displace their space where they interact with this amino acid. They can displace all this hydroxonium ion and hydroxyl ion in the water, thereby displacing, so that thereby breaking this interaction between the water and the protein. So once they break this interaction, then the protein will precipitate out of the solution, or water aqueous environment, then it will precipitate out. The interaction is no longer, or it will no longer interact with water molecule as such. It will have to come out of water in form of what we call a precipitate. So this is just one of the methods where you use an ionic strength to alter with this interaction. pH can be used, can be adjusted to alter with this interaction. Temperature can be used to alter with this interaction. Now, the next thing is you have your proteins being precipitated out. What made them to precipitate out is the presence of this ion. So the next thing is to take a step in order to remove those ions. And the next step is dialysis. In dialysis, you put your protein inside a semi-permeable membrane. So inside this semi-permeable membrane, you introduce it into a proper solution, and that solution, you can have this ammonium ions and the sulfate ions that move out can diffuse out from the proteins into the proper solution easily through the similar force. Meanwhile, the protein, because the semi-permeable membrane contains the smaller force of which the protein cannot be able to move out. So the purification stage. The obtained protein can be purified using quite a number of chromatographic techniques. It can be gel filtration chromatography. And we say in gel filtration chromatography, the separation is based on the site where you have the beads, and some beads contain these openings where the smaller proteins will get in while the bigger molecules will get out faster, and then follow as you are adding the mobile phase, the proteins are getting washed out gradually so that you will be collecting the fractions of this the form of Elliott. And then we have charge or cation exchange or anion exchange chromatography, where the separation is done based on charge. For example, when you are using cation exchange resin, cation exchange resin contain a lot of negatively charged molecules. So those negatively charged molecules can easily interact with positively charged molecules. And even the interaction also here depend on the strengths of the strength of these charges. The molecule can be strongly positive or strongly negative. So if you have a strongly positive molecule, can strongly interact with this negative molecule that was attached to the cation exchange within as a stationary phase ion exchange chromatography. So you will have this molecule to be bonded to, while the highly negative one will move out faster, followed by the intermediate positive, negative, and then followed by a weak negative or slight negative. And then you have those ones neutral. Then you have those ones that slightly bind, meaning those ones that are positively charged but slightly positively charged. Then followed by intermediate, and then followed by strongly positive. Those ones that bonded to the negatively charged ions, which is a strong bonding energy. Then we have an affinity, which is based on the affinity, the love between the proteins. So some proteins, you can design an antibody that can easily bind to a specific protein of interest. Then you put that antibody in as a stationary phase in a column. So when you are passing your sample, this particular protein of interest can actually bind to this antibody. And then the others will be able to have this one will add it later, so that you have your protein collected at the end. So you can also purify through electrophoresis, where you will you have, after in most cases you can do the column chromatography. After the column chromatography, what you expect to be your protein of interest, now you can take it and then run gel. Maybe you have an idea of the size of this protein. So when you run gel, you have a marker, then you'll be able to know whether you've actually isolated the protein of interest. For example, here what we have this as a non-protein. So in between 31,000 Dalton to 45,000 Dalton, you have this. And if your own protein is around 40, let's say 40,000 Dalton, so of course you can say, oh, this is the protein of interest. Or sometimes the protein of interest may not even be pure like this. You can have it and you have included others. Then in some cases, you can isolate this one and continue your work. So, okay. So I will stop here. I will also produce another video where you you're interested in knowing the amino acid composition of this purified protein. Like I told you, you can slice out, you can cut out this protein from the gel and then suspend it in a buffer and then carry out amino acid composition analysis. So in another video, I will talk about how you can get the amino acid composition of a protein. Thank you very much. If you have any question, please forward your question to me anytime, maybe through your classroom.